Hi everyone, welcome to our channel. Today we have a presentation prepared for you on the fascinating world of plant biology, particularly focusing on secondary metabolites that play a crucial role in the plant's life, affecting its growth, development, and response to the environment, and on the phytohormones, which are also known as plant hormones and are the chemical messengers that coordinate the plant's physiological activities. So let's start with the first question, and the first question is, what distinguishes primary metabolites from secondary metabolites? A. Primary metabolites are only found in plants. B. Secondary metabolites are essential for an organism's survival. C. Primary metabolites are non-toxic. D. Secondary metabolites often play a role in plant defense mechanism. The correct option is C, because secondary metabolites often serve roles in plant defense mechanism against herbivores, pathogens, and competing plants. They can be toxic or have antimicrobial properties which helps in the plant's defense. This characteristics is one of the distinguishing features between primary and secondary metabolites. Primary metabolites are involved in the basic survival and function of the cell, while secondary metabolites they have specialized role in the interaction with environment and other organisms. Therefore, option D accurately distinguishes secondary metabolites from primary metabolites based upon their role in defense mechanism, which is a common characteristic of secondary metabolites. Now let's discuss A, B, and C option. The first option is wrong because primary metabolites are not exclusive to plants. They are found in all the organisms, including animals, fungi, and bacteria. Primary metabolites include amino acid, nucleic acids, and sugars, which are essential for basic cellular function and survival. And B is incorrect because secondary metabolites are not essential for organism survival, but rather they help an organism to interact with its environment and other organisms within it. They often play important role in interactions like defense mechanism, attraction of pollinators or competitors with other organisms. Option C is not typically incorrect, but it's like this option is not distinguishable for the primary and secondary metabolites, because secondary metabolites can also be non-toxic. Moreover, some primary metabolites could be toxic if present in improper amount. Let's move to another question. What is the primary role of strigolactones in plant development? A. Promoting root hair growth. B. Inhibiting stem elongation. C. Regulating shoot branching. D. Stimulating flower blooming. So strigolactones are primarily known for their role in inhibiting shoot branching, helping the plant to allocate resources more efficiently. So the correct answer is C, regulating shoot branching. The next question is, which enzyme class is often involved in the biosynthesis of secondary metabolite? A. Transferases. B. Ligases. C. Isomerases. D. Oxidoreductases. So, oxidoreductases are often involved in the biosynthesis of secondary metabolites. They facilitate the redox reaction that can add complexity to these compounds. Next is, what is the key precursor molecule in the biosynthesis of alkaloids? A. Acetyl-CoA. B. Ornithine. C. Shikimate. D. Corismate. So the correct answer is ornithine, because acetyl-CoA it's a central molecule in the metabolism and it's involved in the synthesis of fatty acid and cholesterol and is a starting point for the TCA cycle. However, it's not the key precursor molecule for the synthesis of alkaloids. Shikimate, that is essential for the synthesis of aromatic amino acid, which can be precursor for variety of secondary metabolite. However, it's also not the key precursor for alkaloids. Corismate, it's a crucial compound in shikimate pathway that serves as precursor for synthesis of aromatic amino acid. Like shikimate, corismate is also not the precursor molecule. B option, ornithine is a precursor. It's an amino acid that is not used in protein synthesis, but it is a critical molecule in the urea cycle. and serves as a precursor for the biosynthesis of several alkaloids. Alkaloids derived from ornithine include tropane alkaloids such as atropine, cocaine, and other like pyrrolizidine types of alkaloid. All they are obtained from ornithine. The next question is, how do polyketides differ biochemically from fatty acids in plants? A. Polyketides are branched while fatty acids are linear. B. Polyketides have multiple functional groups while fatty acids have carboxylic acid group only. C. Polyketides are exclusively found in animal cells. D. Fatty acids are synthesized from polyketides. The correct answer is B. Polyketides have multiple functional group while fatty acids have a carboxylic acid group only. Polyketides are synthesized through a process that involves the condensation of small carboxylic acids similar to fatty acid synthesis, but they undergo additional modification that introduce various functional groups. These modifications include cyclization, reduction, and oxidation reaction that generate a wide variety of functional groups and structural diversity. While fatty acids, they typically have a carboxyl group at one end and a methyl group at another, with a long hydrocarbon chain in between. The next is, which biochemical pathway is responsible for the biosynthesis of flavonoids? A. Calvin cycle. B. Schmidt pathway. C. Mevalonic acid pathway. D. Glyoxylate cycle. Most of the people get confused between B and C, but the correct answer is B, because C option, the mevalonic acid pathway, it is also known as mevalonate pathway. It is crucial in eukaryotes, archaea, and some bacteria, but it plays mainly its role in the synthesis of isoprenoids, cholesterol, steroid hormones, prenylation, coenzyme synthesis, dolicol synthesis, or geranyl and farnesyl pyrophosphate synthesis. But this pathway is not responsible for synthesis of flavonoids. While Schmidt acid pathway, it is responsible for synthesis of aromatic amino acid, which are further converted into a variety of secondary metabolites, including flavonoids. The next question is, this question is quite simple. What role do cytochrome P450 enzymes often play in the metabolism of secondary metabolites? A. Hydrolysis. B. Methylation. C. Oxidation. D. Glycosylation. The correct answer is C, because cytochrome P450 enzyme often catalyzes the oxidation of secondary metabolites by modifying their structure and potentially their function. The next question is, which phytohormone is most directly responsible for stimulating seed germination? A. Abscisic acid. B. Auxin. C. Gibberellin. D. Cytokinin. The correct answer is C. Gibberellin. Gibberellin are a large family of plant hormones that play a crucial role in promoting seed germination, stem elongation, flowering, fruit development. It breaks the dormancy of seeds by promoting the digestion of stored food reserves in the endosperm through the synthesis of hydrolytic enzyme, thereby fueling the growth of emerging seedling. So among the all options, gibberellin is the most direct hormone that is responsible for seed germination. While all other hormones that are given here, they play different role in plant growth and or development. For example, in the case of ABA, it inhibits the germination. They are not directly involved in the plant growth and development, but C, that is gibberellin, it's directly and crucially involved in promoting seed germination. The next question is, which enzyme is inhibited by phytohormone ethylene to cause fruit ripening? A. Polygalacturonesis. B. Catalasis. C. ACC oxidases. D. ACC synthesis. The answer is A. Polygalacturonesis. Ethylene actually induces the activity of these enzymes, and as they break down the pectin in the fruit cell wall, leading to fruit softening and ripening. The last question for today is, in what form is auxin typically transported in plants? A. As a gas. B. Bound to proteins. C. In its ionic form. D. In its protonated form. So most of the people get confused between C and D. But the correct answer is D, the protonated form. Although oxygen can lose a proton to become in its ionic form, however, the ionic form of oxygen cannot cross the membrane easily. So the primary form of transport across the cell is the protonated form, not the ionic form. Thank you very much to each and every one of you for tuning in. If you found this information useful or have any more questions, feel free to drop a comment below. We always look forward to your valuable feedback. Don't forget to hit the like button, share this video with your fellows, and subscribe to our channel for more videos. Until next time, keep exploring the microscopic wonders that plant biochemistry unfolds. Thank you and take care.